Hello, my name is Tom Stinchcombe. I'm a medical oncologist who specializes in thoracic oncology. I work at Duke Cancer Institute in Durham, North Carolina. And thank you for having me today. So I think there's been a lot of interest in HER2 mutations. They're generally about 2 to 3% of patients with adenocarcinoma and non-small cell histology. And these are, the majority of them are something called HER2 exon 20 mutations, but mutations occur out of the exon 20. And I think there's a very great similarity between the HER2 exon 20 and the EGFR exon 20 mutations biologically in terms of structure. We've been trying to develop drugs for the HER2 mutations for a prolonged period of time. TDM1 had been shown some promise in a smaller study with a response rate of about 44%. And a number of tyrosine kinases have been developed. Most showed modest activity, and unfortunately, they had a lot of activity against the EGFR on the skin and the gut, so there was a high rate of diarrhea and other EGFR-associated toxins such as rash. And I think that this has been an active area of drug development, and most recently, poziotinib was investigated in this setting. And I think the thing that's going to sort of change the landscape therapeutically is the recent trials with trastuzumab and deruxtecan, which is an antibody drug conjugate that binds to the HER2 protein and delivers a chemotherapy. And a recent trial published in the New England Journal by Dr. Lee and colleagues, the response rate was 55%, and the duration response was around 9 months, and PFS was around 8 months. And the toxicities were what to be expected with an antibody drug conjugate to chemotherapy, some nausea, fatigue, and some diarrhea, as well as some myelosuppression such as neutropenia and anemia or low white cells or low red cells. The unique toxicity is that this can cause something called interstitial lung disease, which is an inflammation of the lungs. And so people have to be very vigilant about this. Patients with HER2 exon 20 mutations were included, as well as other mutations. So I think that it's unclear right now if this drug needs to be just focused on the HER2 exon 20, which were the majority of the patients, and is the benefit really restricted to that. I can tell you in the trial that they did see benefit regardless of the mutation subtype, but I think we're really going to try and make refinements on the HER2 exon 20 HER2 mutations to really try and pinpoint which mutations are more susceptible to this drug. Right now, I would use the drug as it was used in the trial, which was not restricted to the HER2 exon 20 mutations.